I'll record. Okay, I don't see any other hands. So is that everybody who wants to record? Okay. Okay. Do you guys have questions for me before I start reviewing? Nothing?
I have a question.
Uh-huh.
As far as the intrinsic control of blood flow, I just, I guess I was trying to figure out how to differentiate it from sympatholysis. Like, is sympatholysis a version of that?
So sympatholysis you can think of as like the competition between intrinsic control and extrinsic control. So the intrinsic control is the chemoreceptors trying to cause vasodilation. The extrinsic control is the epinephrine trying to cause vasoconstriction. They're both happening at the same time.
Okay.
So the vasodilation will win out. And that's sympatholysis. Is that we're able to get vasodilation even when epinephrine is trying to cause vasoconstriction. That's the sympatholysis.
Okay, cool. Thank you.
Okay, you're welcome. So if I asked you guys right now to close your notes and take out a piece of paper and draw that stroke volume map, would you be able to do it? Have you been working on your stroke volume map? Okay, you got to make sure you guys are keeping up with this information like you did for the first test. Okay, you guys did really well on the first test, but even without all of this online stuff, scores tend to be significantly lower on my second exam. And I don't think it's because the information is harder. I think it's because people don't work as hard on the second exam because they did so well on the first exam. So you did well on the first exam because you worked really hard. So keep that work going as we go into this second exam. Okay, Caroline, go ahead.
So when I was drawing the map, so I know it goes decreased PNS activity to decreased vagal tone, and then it goes to SA node. But like, I was trying, what's the connection between decreased vagal tone and the SA node?
So the vagus nerve, its only job is to act on the SA node. So the more active that nerve is, the more signals that nerve is sending, the slower the SA node will fire. So the vagus nerve acts on the SA node to affect heart rate.
Okay.
So the higher the vagal tone, that means the vagus nerve is more active. It acts on the SA node to slow down the heart rate. If the PNS is withdrawing, you're getting a decreased vagal tone, you're getting fewer signals from the vagus nerve. That acts on the SA node to allow the heart rate to increase.
Okay.
So anything that's affecting the heart rate has to act on the SA node. Okay, let's review a little bit. Kobe, can you please explain intrinsic control of heart rate?
I got to pass on this one.
Okay. Jacob, can you explain intrinsic control of heart rate? You have to unmute yourself.
I'm not sure on that one either.
Okay. Heather, can you please explain intrinsic control of heart rate?
Intrinsic control of heart rate. Let me see. It involves the AV node, the AV bundle Purkinje fibers acting on the heart rate and internal factors.
Okay, so can you explain it in detail?
Yeah, let's see.
Process? Okay, I'll pass it to Svetlana. Can you please explain intrinsic control of heart rate?
It's the involuntary control of the heart. The SA node will initiate electrical signal. It'll contract the atria, and then it sets the beat to 100 beats per minute. The signal will travel down the AV node, and then it will then go down the septum of the heart to the AV bundle and into the Purkinje fibers, causing the ventricles to contract.
Perfect. Okay, so did you guys know that's what I was asking for? Did you connect that term with that process, intrinsic control of heart rate? I also referred to it as cardiac autoconduction. Remember I said cardiac autoconduction and intrinsic control of heart rate are the same thing. Describe how the heart is controlling itself. Okay, I shouldn't have to go through 4 people before I get an answer to that question if you guys are staying up with the material. Okay.
I have one question.
Yes, go ahead.
Sorry. So it goes to the AV bundle first and then the Purkinje fibers? Because I was, okay, I had it the other way around. Okay, thank you.
Okay, James, can you please explain extrinsic control of heart rate?
Okay. Extrinsic control of heart rate. A decrease in PNS activity will raise the heart rate from 60 to 100, and this will also cause a decrease in vagal tone,
Okay.
which will increase the heart rate. And then an increase in SNS activity will raise the heart rate above 100, which causes an increase in the activity of the sympathetic cardiac nerve and also a release in epinephrine.
Good, and both of those things act on the SA node to cause the heart rate to increase. Very
Yes.
good. So we talked about all 3 forms of extrinsic control. Those are 3 different things that are acting on the SA node to change the heart rate. Okay, and we had a graph for that, which you should be able to draw and explain. Everybody okay on that? Okay. Shannon, can you please explain extrinsic control of blood flow?
Is that does that refer also like as you're referring to intrinsic, what the extrinsic would be? I'm a little confused on the question.
So blood flow is controlled by the arterioles, vasoconstricting and vasodilating. So extrinsic control is something external acting on the arterioles to either cause vasodilation or vasoconstriction.
So PNS or SNS. For the PNS, it would be the vagal tone or the vagal nerve.
So that's heart rate. Vagus nerve acts on the SA node, which controls your heart rate. I'm asking about blood flow, which is controlled by arterioles. So remember I said don't get confused between intrinsic control of heart rate and intrinsic control of blood flow. If we're talking about heart rate, we're talking about the SA node. If we're talking about blood flow, we're talking about arterioles.
Okay.
Vasoconstriction and vasodilation. You want to pass it?
Yes.
Okay. Susanna, can you please explain extrinsic control of blood flow?
Is it that like epinephrine causes vasoconstriction?
Yeah, that's it. Epinephrine acts on the arterioles to cause vasoconstriction. Good. Roxanne, can you please explain intrinsic control of blood flow?
So for intrinsic control, basically the only thing that I have is that the arterioles decide to vasoconstrict or vasodilate. Would that be based on like thermoregulation too, or would that be considered extrinsic?
Well, we talked about
that would be for the surface. So never mind.
During
Would
exercise,
it be the sympatholysis then?
Well, this is what I talked about at the beginning with Eddie's question. There, you have to separate the intrinsic control of blood flow from sympatholysis. The intrinsic control of blood flow is related to sympatholysis, but you need to be able to explain intrinsic control of blood flow by itself. So how do the arterioles decide on their own when to vasodilate? This is happening at the exercising muscle, remember? You're exercising, you secrete epinephrine. Epinephrine is going around everywhere trying to cause vasoconstriction. But at the exercising muscle, we need vasodilation. So how do we make that happen?
Would it be because of the chemoreceptors?
Yes.
So I'm gonna have to pass it. I can't put
Okay.
them all together.
Anyone want to volunteer for this one? Ismail, you want to do it? You got to unmute yourself.
Okay, so basically, because of the vasoconstriction, it happens everywhere except in the vessels near the skin.
That's true.
So, whereas it causes vasodilation for thermoregulation.
That's epinephrine, which is extrinsic control.
Mm.
I'm asking for intrinsic control. Specifically, how do we get vasodilation at the exercising muscle?
Are you looking for sympatholysis?
Yes, but the details of how that vasodilation is triggered.
Is this where the
O2? Oh,
sense the
Yes,
local
Caroline. Chemoreceptors sense what?
the local conditions at the exercising muscle, which
Yes.
triggers vasodilation and
right.
artery.
Yes. What are those local conditions?
Lower O2, higher CO2, higher, oh, I just put HLA. I can't remember what that stands for.
Lactic acid.
Oh, thank you. Higher hydrogen, lower pH, and higher temperature.
Yes. So you're exercising, and those are the local conditions of the exercising muscle, right? Because the muscle is consuming oxygen, so oxygen levels are low. The muscle is producing carbon dioxide and lactic acid, so those levels are high. Okay, so you have all these conditions at the exercising muscle that's sensed by the chemoreceptors that will trigger vasodilation. And that's what brings the increased blood flow to the muscle. Okay, so the sympatholysis is, like I said before, the epinephrine is trying to cause vasoconstriction, the chemoreceptors are trying to cause vasodilation. Both things are happening at the same time. So who's going to win that battle? It's got to be the chemoreceptors so that we can vasodilate, so that we can increase blood flow to the muscles. Okay, but you've got to understand extrinsic and intrinsic control of blood flow. Blood flow is controlled by arterioles. So if we're talking about extrinsic and intrinsic control, it's got to be happening at of blood flow, it's happening at the arterioles. Extrinsic control is epinephrine acting on the arterioles. Intrinsic control is the arterioles controlling themselves, which is through the chemoreceptors. Okay, everybody okay with that? That seems to be a kind of a sticking point for you guys. Do you have any other questions on the extrinsic and intrinsic control of heart rate or blood flow? And the other term for intrinsic control with heart rate was cardiac autoconduction. The other term for intrinsic control of blood flow was arterial autoregulation. Okay, Harrison, can you please give a definition and a formula for stroke volume?
Stroke volume, the amount of blood pumped by the left ventricle per beat, and that delivers blood to the rest of the body and is controlled by the left ventricular myocardium.
Good. Formula?
Cardiac output equals heart rate times stroke volume.
But I asked you to find stroke volume, right? So what's the formula for stroke volume?
I don't think I have that down right
Okay,
here.
London, what's the formula for stroke volume?
Stroke volume equals end diastolic volume minus end systolic volume.
Good. While you're there, go ahead and define end diastolic volume and end systolic volume.
Okay. End diastolic volume, amount of blood in left ventricle at the end of distally. End systolic volume, amount of blood in left ventricle after systole.
Good. David Gregorian, what is end diastolic volume?
End diastolic volume is the amount of blood left in the ventricles after contraction.
Good. Brianna Hernandez, what is end systolic volume?
End systolic volume is when they are contracting and they should be lower.
So what's your definition of end systolic volume?
Sorry. The amount of blood in the left ventricle at the end of systole.
Yes. Alexis Harrington, what is systole?
Is it contraction?
Yes, contraction of what?
Of the contraction of the heart.
The whole heart doesn't contract at the same time. Is it the atria or the ventricles?
Oh, the ventricles.
Ventricles, good. Brianna Chavez, what is diastole?
Contracting of the atria.
Good, so the ventricles are relaxing. So we're mostly going to focus on what's happening at the ventricles, because that's what we're most concerned with. Okay. Guillermo, can you please give me the short and expanded formulas for blood pressure?
No, I can't. I don't remember right now.
Vanessa K.
The short one is cardiac output times peripheral resistance, and the long one is heart rate times stroke volume times length times volume times radius to the fourth power. Divided by radius to the fourth power.
Yes, and it's length times viscosity divided by radius to the fourth. Good.
I'm sorry, what was the short version of it?
Blood pressure equals cardiac output times peripheral resistance. And then we can expand cardiac output to heart rate times stroke volume. We'd expand peripheral resistance to length times viscosity over radius to the fourth. Okay, so then we took all of that and expanded on it, right? Talked about the things that cause the increase in heart rate during exercise, the things that cause the increase in stroke volume during exercise, the things that cause a decrease in resistance during exercise. Let's talk about acute control of blood pressure, right? So what I'd like to do is I'm going to put my whiteboard up. Clear it. Okay, so I want to review this drawing, but you guys all have it drawn out already, so I don't want you to just look at that because that's passive. So what I'd like you to do is close your notes so you don't have that drawing in front of you and see if you can help me recreate this drawing and make it more active. Okay, so this drawing is acute control of blood pressure. How do we get blood pressure to increase during exercise? Okay, so it's going to start with exercise. And then what is the sensory receptor that's going to control blood pressure?
SA node.
SA node is heart rate. And it's not a sensory receptor.
Baroreceptor.
Baroreceptor is the sensory receptor that controls blood pressure. It's going to send its information to the brain, and the brain is going to respond in two different ways. What are the two different ways?
Parasympathetic and sympathetic.
Okay, so the parasympathetic, is it going to increase or decrease its activity?
Decrease.
Decrease.
Yes, because we're exercising. So decrease parasympathetic nervous system activity through what? How is the PNS going to act here?
Vagal nerve. Decreased vagal tone.
Decrease vagal tone, which acts on SA node. And how does the SA node respond to a decrease in vagal tone?
Increases heart rate.
Increased heart rate, which will cause
An increase in cardiac output.
increased cardiac output, which will then cause increased blood pressure. Okay, so I'm going to go back and draw my arrows in here. Decrease in PNS activity will decrease vagal tone, act on the SA node, which I'm going to box as my effector, will increase heart rate, will increase cardiac output, will increase blood pressure. Okay, so would it be okay to put increased stroke volume here? Does an increase in heart rate cause an increase in stroke volume? Does it?
Yes.
Yeah. Yes.
How does an increase in heart rate cause an increase in stroke volume?
I want to say no, but I don't know why.
What does stroke volume depend on?
Increase in contraction strength.
Yes. What's the formula for stroke volume?
In this stroke volume equals in systolic
Volume.
volume
Minus systolic volume.
minus systolic.
Is heart rate in there somewhere? Does stroke volume depend on heart rate?
No, it does not.
Sorry.
No. So an increase in heart rate is not going to cause an increase in stroke volume. If anything, an increase in heart rate would cause a decrease in stroke volume because the heart is beating so quickly, you don't have enough time to fill the ventricle. So if anything, it would cause a decrease. So we're not going to, so stroke volume doesn't belong there. Okay, so don't just connect heart rate and stroke volume. If heart rate's increasing, stroke volume is increasing. Heart rate affects cardiac output. Stroke volume affects cardiac output, but an increase in heart rate is not going to affect the stroke volume. Okay. Okay, what's the other way the brain can respond here?
Sympathetic system.
Increase or decrease activity?
Increase.
Increase.
Increase sympathetic nervous system activity. How many different ways?
Two.
Two. What are they?
Epinephrine and
cardiac nerve.
Cardiac nerve. Okay. What does the sympathetic cardiac nerve act on?
SA node.
SA node, which causes increased heart rate, cardiac output, and blood pressure. And how many different branches do we have coming off of epinephrine?
3.
What are they?
The SA node.
SA node.
The arterioles.
Arterioles and
Left ventricle or no.
That's right.
Ventricular myocardium.
Left ventricular myocardium. Good. Does it matter which one I put first?
No.
Yes.
Yeah.
Yes, yes it does. Sorry.
Yes, it does.
Which
so you
one
don't
has to go first?
The SA node.
SA node has to go first because I can't cross lines. So if I do arterioles here and have a line that goes all the way to blood pressure, then my next line, it would have to cross over it to get to SA node. Okay, I'm moving my cursor as if you guys can see it. Okay.
We can see the cursor.
You can see the cursor?
Yeah.
You can? This?
Yeah.
Oh. Well, that's good to know. Okay. So for the other two branches of epinephrine, it doesn't matter which order because they're each going to have their own pathway. Okay, so what do you want to put next?
Left ventricular.
Okay, left ventricular myocardium. And how does that respond to epinephrine?
Increase contraction strength.
Okay, and what does that cause?
Increase stroke volume.
Okay, stroke volume, and what does that cause?
Increase in blood pressure.
Cardiac output.
Increase in cardiac output. Good, so let me put my arrows here. All right, that one's going here. Okay, so could I put increase in heart rate here? Would that make sense?
No.
No.
No. An increase in stroke volume is not going to cause an increase in heart rate. Those are going to be separate here. Okay, and our third branch from epinephrine?
Arterioles.
Arterioles. How do the arterioles respond to epinephrine?
Vasoconstriction.
Vessel, yeah.
And what does that cause?
Decrease in vessel radius.
Vessel radius, which causes What comes next?
An increase in peripheral resistance.
Increase in which causes
The increase in blood pressure.
increase in blood pressure. So let me connect these. Okay, I need to identify my effectors. And I will just box blood pressure since that's our goal. Okay, so everybody okay with this map? Okay, so let me ask. Marcia, can you please explain one way epinephrine contributes to increases in blood pressure during exercise?
Can we open our notes now?
Yes.
Can you repeat the question, Professor?
Yes. Can you please explain one way epinephrine contributes to increases in blood pressure during exercise? That's based on this map.
Epinephrine can contribute to the arterioles, which is the effector, which vasoconstriction, which decreases the radius, which increase the peripheral resistance, which increase the blood pressure.
Good. Armin, I almost called on you. Alex, can you please explain one way epinephrine contributes to increases in blood pressure during exercise?
It will increase heart rate, which increases cardiac output, which increases blood pressure.
Okay, how does epinephrine increase heart rate?
Increase in SNS activity.
Okay, but you need to say epinephrine acts on the SA node. Because in order to increase heart rate, you have to act on the SA node.
Okay.
So you're just missing that detail.
Okay.
Okay. Alejandro, can you please explain one more way? I can't see you, so I don't know if you are responding or not. I will move on. Carolina, how do you say your name? Say it again, please. Can't hear you.
Carolina?
Carolina. Okay, thank you, Carolina. Could you please explain how epinephrine increases blood pressure?
Through the left ventricular myocardium,
Okay.
which increases contraction strength,
Mm-hmm.
which increases stroke volume and cardiac output.
Perfect. Thank you. Let's see. Jacob, can you please explain how the parasympathetic nervous system is involved in increases in blood pressure during exercise?
Can you repeat that again?
Yes. Please explain how the parasympathetic nervous system is involved in increases in blood pressure during exercise.
Can you repeat that one more time?
Explain how the parasympathetic nervous system contributes to increases in blood pressure during exercise. So all you have to do is follow that pathway on the map we just drew.
So the decrease of parasympathetic, how you say it?
Parasympathetic.
The parasympathetic activity decreases the vagal tone,
Mm-hmm.
which
You just say acts on the SA node.
acts on SA node, which increases the heart rate, which increases the
Cardiac output.
cardiac output, which increases
It's blood pressure.
Blood pressure, my bad.
Yes. So you guys understand how to use this map to help you organize answers to those kinds of questions, yes?
Yes.
Okay, any questions on any of that? Okay, then we talked about venous return mechanisms. Nick, can you please just name the 3 venous return mechanisms? You don't have to explain them. Just name them.
The skeletal muscle pump, the lung pump,
Respiratory pump.
Respiratory pump. And I can't remember the last one. I'm trying not to use my notes, but let me think.
That's okay. Putting your thumb over half of the hose makes the water come out faster. So as we make the vessel narrower.
Oh, vasoconstriction?
It's vasoconstriction, yes.
Okay.
Good. And then we talked about components of the blood and hematocrit. Cindy, what's the formula for hematocrit?
Is it 45 for formed elements and 55 for plasma?
That's normal hematocrit, but what's the formula for hematocrit?
Formed elements over total blood volume.
Right. So the more of your total blood volume that's made up of formed elements, the higher the hematocrit, which means your blood is thicker, right? So as your blood gets thicker, hematocrit goes up, which means viscosity goes up. Viscosity is related to peripheral resistance. So think about peripheral resistance as resistance to blood flow in your extremities. So if hematocrit is high, viscosity is high, resistance is high, which means it's going to get it's going to be more difficult for blood to flow to the muscles if it's thick. So we like to have high levels of red blood cells because that increases our oxygen carrying capacity. The red blood cells have the hemoglobin that carries oxygen. So we like to have high levels of red blood cells so we can carry more oxygen, but we don't want our blood to be thick. We want our blood to be thin. So ideally we have high levels of red blood cells, but also high levels of plasma so that our hematocrit stays low, so that the blood stays thin, so we don't have resistance to blood flow. Okay, and that's exactly what happens with endurance training, as we'll talk about again after the midterm. You increase your red blood cell count so you can carry more oxygen. You also increase your plasma so that your hematocrit stays low, and then you have the best possible situation. Okay.
Is there not a danger to having like your blood too thin?
Having your blood too thin, I think the issue with that is that you can bruise really easily. Okay. And
it's not bleeding out if the
yeah, and you can bleed. You would bleed out faster if your blood was really thin. Yeah, but also if your blood is thin as a result of low formed elements, then that's not a good thing, because then you can carry less oxygen, like anemia. A person who's anemic would have thinner blood, but because they have fewer red blood cells, so that's going to be a problem. Yeah. Okay, we talked about altitude training in here, yes or no? Okay, talked about altitude training triggers your body to produce more red blood cells. We talked about blood doping, yes? We didn't talk about blood doping. Okay, so blood doping is another way to increase your red blood cells. This is what we've heard about the cyclists doing to improve their performance. So blood doping is when you take out some of your blood and you put it in the freezer so it stays alive, and then your body's going to replace those red blood cells. Okay, your body's constantly replacing the red blood cells that die off. So you're always making them. So your body will replace what you take out, and then right before your competition, you inject them back in. So you have extra red blood cells. If you have extra red blood cells, you have extra hemoglobin, which means you can carry more oxygen, which is going to improve your endurance performance significantly. Okay, but again, that only works if the person is trained, so they have the mitochondria and the myoglobin and all that. And it's all natural. They're not taking any drugs. It's their own blood that they're putting back in, but it's illegal because it gives you an advantage over people who are not doing it. And hold on one second. They have to, when they're injecting that, they can't just inject the red blood cells. They have to inject plasma as well, because if they just inject the red blood cells, it increases your red blood cells, but it increases your hematocrit, which is going to be a detriment to your performance. So you've got to have the plasma levels and stay really well hydrated as well. Okay, someone have a question?
Yeah, I was going to ask, how do they catch that? Like, is there like a test to see old red blood cells versus new ones?
That's a good question. It would be very difficult to detect because it's all natural.
Right.
It would just be, they must do like, it must be more than a one-day test. Like they might test them a couple days apart, because it wouldn't be normal to have that significant of an increase in red blood cells from day to day unless it was injected.
Yeah,
So that's my guess on how they do it. But. Okay, any other questions on that?
Isn't there like a certain amount of blood that you're supposed to have? Like, can you re-inject back too much, or is that? I'm curious.
Yeah, it's a small amount that they do. I don't know what the amount is, but yeah, it's not a lot.
So just to go over really quick, blood doping is when you take out your red blood cells, freeze them, and then after training, you inject them back into your body.
Well, right before the competition. This person is already trained, because this is elite athletes that do this, right? So the person is already well trained, but maybe a month or two before the event, they might take the blood out, and they're not just taking out red blood cells, they're taking out the blood cells out in the plasma. And then in that amount of time, their body will replenish that, they'll be back to normal levels, and then right before the event, they would inject it in.
So it's whole blood they're taking out.
Whole blood.
Whole blood, okay.
Yeah, if they just injected the red blood cells, their blood would be too thick. Okay, so that. Okay, so I have one more topic in this chapter. So for this one, imagine that it's the middle of August and they've just let us out of quarantine, and you go to a party and you eat an entire pizza and you drink an entire 6-pack of beer, and the next day you feel really gross and mad at yourself, and you decide you're going to go to the gym and work out. It's 105 degrees out, and you go to the gym and the air conditioning is broken and you forgot your water, but you're going to work out anyway because you're determined, and you're going to do an hour of cardio. You're going to do an hour-long run on the treadmill, which is normal for you. You do it all the time. This is imaginary. Okay, so imagine that's a normal workout for you. Okay, so you go to the gym, it's 105 degrees out, you don't have your water, the air conditioning is broken, all the windows are closed, it's really hot in there. You're running on the treadmill at your comfortable pace. Let's say that's 6 miles per hour. Okay, so, and by the way, this was something that a lot of people had trouble with on the Kahoot quiz that I gave you with steady state. Steady state has to be aerobic. So remember, steady state is when your oxygen supply meets your oxygen demand. You're getting enough oxygen. By definition, that's aerobic. If you can't be in steady state and be anaerobic, because in anaerobic, you're not getting enough oxygen. Your requirement for oxygen is above what you're consuming. So in order to be in steady state, you have to be working aerobically underneath your lactate threshold. Okay, so in this example, you're running at 6 miles per hour, you're at steady state. So you're working aerobically, it's a comfortable pace for you, you can maintain that for an hour, you do it all the time. Okay, so if you're maintaining your pace at 6 miles per hour, what should be happening to your heart rate? Should it be increasing, decreasing, or staying the same?
Staying the same.
Staying the same. So in other explanations, in other discussions that we've had heart rate increasing, like the graph where we had intensity on the bottom and heart rate on the y-axis, and we said heart rate is increasing. That's heart rate increasing while intensity is increasing. So that's if you're going 4 miles per hour, 5 miles per hour, 6 miles per hour, 7 miles per hour, your intensity is increasing, your heart rate would be increasing. If we're keeping a steady intensity, your heart rate should be steady. Okay, so you're in this hot environment, you're jogging at 6 miles per hour. Let's say your heart rate is at a steady 140. Okay, after 20 minutes, 30 minutes in, your heart rate starts to drift up. 145, 150, 155, but you haven't changed the speed on the treadmill. Okay, so this is not normal. Normal would be if your intensity stays the same, your heart rate stays the same. In this situation, your intensity is staying the same and the heart rate is increasing. So this is called cardiovascular drift. Okay, I'm going to put my whiteboard up. cardiovascular drift. So cardiovascular drift is when there's an increase in heart rate without an increase in intensity. So this is abnormal. Normal would be for heart rate to stay the same, but heart rate is creeping up, it's drifting up, cardiovascular drift. So it's important that in the example I gave you, you're in a hot environment. Okay, so cardiovascular drift tends to happen in a hot environment with prolonged exercise. Okay, so the issue that's happening here is if you're exercising in a hot environment, you're going to have increased blood flow to the skin. If you have increased blood flow to the skin, you're going to have decreased venous return. Okay, you have more blood going to the skin, that means you have less blood going to the heart. If you have decreased venous return, it's going to end up decreasing your stroke volume. If you have less blood coming to the heart, you're gonna have less blood coming out of the heart. Okay, this increased blood flow to the skin is also going to cause, let me just draw a line over here. Increased blood flow to skin is going to cause decreased venous return and decreased stroke volume. It's also going to cause sweating. If you're sweating, that's coming from your blood plasma. Okay, so that increased sweating is going to decrease plasma levels. If you have less plasma, you're also going to have less venous return and less stroke volume. So basically what's happening, I know this looks like a bit of a mess. Basically what's happening is you're going to have less stroke volume, okay? Because you're sweating, so you have less plasma, so you have less total blood volume. Less blood going to the heart means less blood out of the heart. You're redirecting blood flow to the skin, so we have less blood going to the heart. Decreased blood to the heart means decreased stroke volume. So the issue is if we're maintaining our intensity, we need cardiac output to stay the same. Okay, if we're running at 6 miles per hour, we know what the oxygen requirement is for a 6 miles per hour run. We could calculate that, right? So cardiac output needs to stay the same to deliver that same amount of oxygen. Cardiac output depends on heart rate and stroke volume. So in this situation, we need cardiac output to stay the same, but stroke volume is decreasing. So what has to happen to heart rate in order to maintain cardiac output?
To increase.
It has to increase. So we're in a hot environment. All of these conditions are causing our stroke volume to decrease. If we want to maintain our intensity, we have to maintain our cardiac output. So heart rate has to increase to help us maintain our cardiac output. So that's what's causing the cardiovascular drift. Okay, I'm gonna go over all that again. So this is happening in a hot environment. So remember I said it's 105 degrees, you're in the gym, there's no air conditioning, there's no windows, you forgot your water. So that's a big part of the problem here. If you're drinking water, this wouldn't be as likely to happen. So this is related to dehydration. This is what's going to happen as you become dehydrated. Your heart rate is going to start drifting up. Okay, so because you're dehydrated, this is what's causing you to be dehydrated. You're in a hot environment. You're going to increase blood flow to your skin, which is going to cause you to sweat. That sweat is coming from your plasma, so your plasma level drops, which means you have less blood flow going to your heart. That's a decreased venous return. Is going to decrease stroke volume. The less blood flow you have going into your heart, the less blood you can get out. Also, the more blood we redirect to the skin, the less blood we're directing to the heart. Decreased venous return causes decreased stroke volume. So if stroke volume is decreasing, we have to increase the heart rate in order to maintain the cardiac output. Okay, everybody okay with that? Tends to occur in a hot environment because it's related to dehydration. Any questions on cardiovascular drift?
Can you say that one more time?
The whole thing?
The whole thing.
Okay. Okay, cardiovascular drift is an increase in heart rate without an increase in intensity. It usually happens in a hot environment with prolonged exercise. So if you're in a hot environment, you're going to be redirecting a lot of blood flow to the skin. That's going to cause a decreased stroke volume in two different ways. One way is just in the fact that you're redirecting blood flow to the skin, less blood is going to the heart. That's going to decrease stroke volume. The other reason increased blood flow to the skin decreases blood flow to the heart is because you're sweating and you're losing plasma. So you have less blood, period. Less blood will go to the heart, less blood will come out of the heart. So if you're trying to maintain your intensity, you have to maintain the cardiac output. Cardiac output depends on heart rate and stroke volume. So if you're trying to maintain your cardiac output and stroke volume is dropping, your heart rate has to increase to make up for that. So you can maintain your intensity. We good?
Yes, thank you.
You're welcome. Okay, so that's the end of chapter 6. As far as the quizzes go, I know there's a lot going on right now. You guys have a lot to worry about. Everybody has extra responsibilities, different responsibilities. Everybody's schedules are off. So I know a lot of people are missing the quizzes. So I want you to relax a little bit about that. I'm going to be reopening the chapter 5 quiz, those of you who missed it. The chapter 5, 6, and 7 quizzes I will leave open until the day of your midterm. Okay, they will close at 8 a.m. the day of your midterm. So just go back and make sure you get all of those done. Okay, there's a due date on there right now. I will go back and I will extend it to the day of your midterm. But make sure you take it by then, because after that I won't want to reopen it. Okay, so if you guys don't have any questions on chapter 6, I'm going into chapter 7, which is respiratory control. No questions? Okay, I'm gonna put my whiteboard back up. This.
I actually have a question for chapter 6.
Go ahead.
I'm still kind of confused about because like in the study guide it says the why is it dangerous when hematocrit increases and about viscosity decreasing.
Yes, so if hematocrit increases, viscosity increases, that causes an increase in peripheral resistance, which causes an increase in blood pressure. So that can trigger a heart attack. So if someone who has some risk factors for a heart attack becomes dehydrated and they're exercising and they continue exercising, that could trigger a heart attack because hematocrit goes up. All right, respiration. We are going to separate into 4 separate parts. Okay, first one is going to be pulmonary ventilation. Pulmonary ventilation is moving air in and out of your lungs. Pulmonary ventilation, moving air in and out of the lungs. Number 2, pulmonary diffusion is the gas exchange at the lungs. So that will be oxygen that you're breathing in, that oxygen moving into the blood, carbon dioxide moving out of the blood into the lungs so you can expire it. That's your gas exchange. Okay, oxygen moving from the lungs to the blood, carbon dioxide moving from the blood to the lungs. We'll go over that again later. Number 3 is transportation. That's delivering the oxygen to the muscles. Transportation is delivering oxygen to the muscles. And then number 4 will be capillary diffusion, which is the gas exchange. Okay, so 4 parts of respiration. The first 2 together are considered external respiration. Capillary diffusion is considered internal respiration. And then transportation is the link between the two. The link between the external and internal respiration. Okay, so you breathe air into your lungs, that's pulmonary ventilation. The oxygen moves into your blood, that's pulmonary diffusion. That blood goes to the muscle, that's transportation. The oxygen gets dropped off at the muscle and goes into the muscle, that's capillary diffusion. Okay, then that blood is going to go back up through the right side of the heart into the lungs again, and the process starts over. Okay, so this chapter is going to go through each of these 4 parts in detail. Okay, I'm going to try to get through the first, well, we'll get through the first one and part of the second one right now. And the rest of it we'll do next week. Okay, so I'm going to go detail into pulmonary ventilation. So I'm going to clear this, start over. Okay, so pulmonary ventilation. Moving air in and out of the lungs. So we separate that into 2 parts, inspiration and expiration. Inspiration is bringing air into the lungs. Expiration is moving air out of the lungs. So together they make up pulmonary ventilation, moving air in and out of the lungs. So the way that works is you have your diaphragm muscle, which is horizontal, separates your thoracic cavity from your abdominal cavity. And when it's resting, when it's relaxed, it's curved upwards, which compresses your thoracic cavity. Okay, so if you want to inspire, if you want to breathe in, you contract your diaphragm. When you contract your diaphragm, it flattens out. When it flattens out, it compresses the abdominal cavity and allows the thoracic cavity to expand. That's where the lungs are. So the lungs are going to expand. Okay, as the lungs expand, the pressure inside the lungs is going to drop. Okay, there's an inverse relationship between pressure and volume. Volume being the amount of space that you're occupying, pressure being how tightly the atoms are pushed together. Okay, so if we were to take all 45 students in this class and smash them into a closet, we'd have low volume, a small amount of space, we'd have high pressure, you guys would be pressed against each other. Okay, so there's an inverse relationship between pressure and volume. The volume went down, the amount of space we had went down, the pressure went up. Okay, if we were to take all 45 of you and release you into room 159, that big room, you guys could spread out. Okay, so the volume is increased, the amount of space we have is increased, therefore the pressure is decreased. You guys don't have to be close together anymore. Okay, inverse relationship between pressure and volume, that is Boyle's law. Let me write that here real quick. Change the color. Boyle's law says there is an inverse relationship between pressure and volume. Inverse relationship means that one goes up, the other goes down. Boyle's law. Okay, so you contract your diaphragm, it flattens out. That allows the thoracic cavity to expand, which allows the lungs to expand. So that's an increase in volume, which means there's a decrease in pressure. So now there's a difference in pressure between the outside air and the air inside your lungs. Pressure gradient means there's a difference in pressure. So we've created a pressure gradient. The air is always going to move down the pressure gradient from areas of high pressure to areas of low pressure. Okay, so we've expanded our lungs, pressure dropped. Now there's high pressure in the outside air, low pressure inside the lungs, so the air is going to move into the lungs, down the pressure gradient. That's how we get air into our lungs. So a lot of people think the air coming into our lungs is what causes the lungs to expand. It's not true. You contract your diaphragm, that allows the lungs to expand, which creates a pressure gradient. And then the air is going to move down the pressure gradient. So the lungs are going to expand first, and then the air is going to come in. Okay, so is inspiration active or passive?
Active.
Active. What makes it active?
You're doing it. It's no.
Yeah,
That's not how we define active. Eddie, how do we define active?
It uses ATP.
Yes, it requires a muscle contraction, which uses ATP. What muscle did we contract for inspiration?
The diaphragm?
The diaphragm.
Rectus abdominis?
Rectus abdominis is not involved in inspiration. It can be involved in expiration, which we'll get to in a second. So, diaphragm in its resting position is curved upward, which compresses the lungs. So when you're ready to inspire, you contract your diaphragm, it flattens out, lungs expand, increase in volume, decrease in pressure, creates a pressure gradient, high pressure on the outside, low pressure on the inside, air moves into the lungs. Active process. Okay, now when you want to expire, all you have to do is relax your diaphragm. You relax your diaphragm, it goes back up into its resting position, which is curved upward, which compresses the thoracic cavity, which compresses the lungs. That's a drop in volume. What happens to pressure? It increases. Drop in pressure, drop in volume, increase in pressure. Now we've created a pressure gradient, but now the high pressure is inside our lungs and the low pressure is in the outside air. So the air is going to move out of the lungs, down the pressure gradient. Is expiration active or passive?
Passive.
Passive.
It's passive. All you had to do was relax your diaphragm, creates a pressure gradient and causes you to exhale. You can make expiration active. For example, if you want to blow out some candles, you forcefully exhale, you can. All you have to do for that is to use additional musculature to help you compress your thoracic cavity. So that's where the rectus abdominis would come in. The rectus abdominis can help you compress. Your intercostals can help you compress your thoracic cavity. So you're compressing your lungs, which creates more of a pressure gradient. As you decrease the volume in your lungs, pressure goes up. More of a pressure gradient means more air is going out. Okay, so inspiration is active. Expiration is normally passive, but can become active if you involve additional musculature. Okay, everybody okay on that?
So does that mean that your diaphragm can become sore?
Diaphragm doesn't normally become sore. You can get a cramp. When you get a cramp in your side, that's usually your diaphragm muscle not getting enough oxygen. Yeah. Okay.
When you have like a sharp pain on your side, would that be your diaphragm?
Usually, yeah. That's the. I'm referring to that as a cramp. I don't know if you're referring to something different, but like as you take a deep breath, usually if you're exercising in a high intensity, you take a deep breath and you get that sharp pain in your side. That's usually your diaphragm.
Aren't hiccups a diaphragm spasm? Like when your diaphragm is spasming?
Cindy, it's really hard to understand you. I'm sorry.
I don't know why. Aren't hiccups a diaphragm spasm when you have hiccups?
Yes, the diaphragm spasming when you have hiccups.
Yeah. Okay.
Yep. Okay, so in the lab, back when I used to teach the lab, we used to measure lung volumes. I don't think they're doing it anymore, but we used to measure lung volumes. So we'd hook you up onto a mask that's going to measure the air going in and out. We'd have you take a couple of a couple of normal breaths, and then you take a maximal inhale, take in as much air in your lungs as you can, and then do a maximal exhale. Expire as much air as you can. So we're trying to measure how much air you can move through your lungs. Okay, so I'm gonna put my whiteboard back up, and I'm gonna clear this. So when we do that, so it would be drawing a graph as you're breathing. So as you inspire, the curve would move up, and as you expire, the curve would move down. So it looks like this. You do a normal breath, and then a normal breath, and then you do a maximal inhale, followed by a maximal exhale, followed by a normal breath, and a normal breath. Okay, so I'm going to draw a line to mark off where my max inhale was. I'm going to draw a line to mark off where my max exhale was. I'm going to draw a line across the top of my normal breath. This is challenging. And I'm going to draw a line across the bottom of my normal breath. Okay, and then I'm going to mark off the different and label the different lung volumes. So during this normal breath right here, this is called tidal volume. Tidal volume is the amount of air you move through your lungs during a normal breath. Tidal volume is the amount of air you move through your lungs during a normal breath. But you could inspire more air than that if you chose to, right? So that's the amount of air you could inspire beyond your normal inspiration. That is inspiratory reserve. So inspiratory reserve volume is the amount of air you could inspire beyond a normal inspiration. It's your inspiratory reserve, that makes sense. If we put those two, combine those two together, whoops, it's the wrong color. Combine those two together, that is your inspiratory capacity. So inspiratory capacity, say inspiratory capacity is tidal volume plus inspiratory reserve volume. You see how that works? Inspiratory capacity is a combination of the tidal volume and the inspiratory reserve volume. Okay, everybody okay with that? Raise your hand if you're not.
Can you repeat the definition for IRV again?
Inspiratory reserve volume is the amount of air you could inspire beyond a normal inspiration. Okay, so you can imagine sitting there doing your normal breathing, and then I ask you to take a deep breath. The amount of air you take in maximally is more than the amount of air you take in a normal breath. Okay, so the difference between the two would be your reserve. Okay, then you could expire more air than you do in a normal breath, right? So that would be here. And that would be expiratory reserve volume. Expiratory reserve volume is the amount of air you could expire beyond a normal expiration. Expiratory reserve volume is the amount of air you could expire beyond a normal expiration. When you do a maximal expiration, do you think you're getting all the air out of your lungs, or do you think there's still some left in there?
Still some.
There's still some left in. We try to get all of it out, but there's always a little bit left in. So I need to draw another line here. Whoops, that should be blue. You should draw another line here to represent air that's still in the lungs after a maximal exhale. This is called residual volume. Residual volume is the amount of air in your lungs after a maximal expiration. Residual volume is the amount of air in your lungs after a maximal expiration. So we don't want that to be a lot. We want to get as much air out as possible, but there's always a little bit left in there. That's residual volume. Okay, if we combine those two, that's called functional residual capacity. functional residual capacity is expiratory reserve volume plus residual volume. Makes sense, yes? Okay, I have two more lung volumes I'm going to put on here. From the top of your maximal inspiration to the bottom of your maximal expiration. That is your vital capacity. Vital capacity is the maximum amount of air you can move through your lungs in one breath. Vital capacity is the maximum amount of air you can move through your lungs in one breath. So if you take a maximal inhale and a maximal exhale, maximum amount of air you can move in one breath, vital capacity. So we can write an equation for this. Vital capacity equals, what would it be?
IRV plus TV plus ERV.
That would work. IRV plus TV plus ERV, but can we simplify that a little bit?
IC plus.
Yes, because the inspiratory capacity is the inspiratory reserve plus the tidal volume. So you could put both of those in there, but it's simpler to combine them. Inspiratory capacity plus
FRC.
It's not FRC because vital capacity does not include residual volume.
ERV. ERV.
ERV. Okay, do you understand why if you put FRC that would be wrong? FRC includes residual volume, vital capacity does not.
Yeah, I see it.
Okay, but the next one does. So this one is going to be basically vital capacity, but also including the residual volume. So this one is total lung capacity. Total lung capacity equals what?
IC plus ERV plus RV.
Okay, that would work. Inspiratory capacity plus expiratory reserve plus residual volume would work. Can you give me a simplified version?
VC plus
RV.
RV.
VC plus RV would work. So there are a few different equations you could put together here that would make sense. Any of them will be correct as long as they include the correct things. Okay, so there are 8 lung volumes on here. You guys need to be able to draw and label a graph like this, although I'm not sure on the online test you'll be able to draw. So it's more likely that you might just need to identify them or define them and not actually draw it. I haven't figured that out yet. Okay, but you need to know these lung volumes. There is a printout of this on Canvas. If you go, I'm pretty sure it's under the chapter 7 module. So if you go on Canvas into modules and scroll down to chapter 7, there should be a link that says lung volumes, and it's a handout of this, and it's really clear in how it shows the relationship between the different lung volumes. So I think it's really helpful. This information is in the book, but I think the book only has like 4 of the lung volumes, and I gave you 8. So make sure you know all 8 of them.
These are on Canvas.
I'm going tomorrow.
Okay.
I'll be back at like 7:15.
Okay. Is it, yes, it should be on Canvas in the chapter 7 module. Okay, other questions on this? It's 1:42 already. How do I not get through as much information in this class as I do in the other ones? Okay, real quick, I'm going to start the next one, which is pulmonary diffusion. Okay, I'm going to clear this. You can take a screenshot if you want, but you also have the handout. Okay, I'm just going to start pulmonary diffusion real quick. Pulmonary diffusion was the gas exchange in the lungs. So this is between the alveoli and the capillary, right? Because as your lungs branch out, your bronchioles branch out, the smallest part is the alveoli. That's what I'm most concerned with. The alveoli is covered with capillaries. And that's where the gas exchange is taking place. So we do green. So green. So if this is the wall of the alveola has a membrane. And then if this is the wall of the capillary also has a membrane. Okay, so we have the alveolar wall, and we have alveolar basement membrane. Okay, and then we have the capillary wall and the capillary basement membrane. Okay, all four of these things together make up the respiratory membrane. Respiratory membrane is what the gases need to diffuse across. Okay, so 4 parts of the respiratory membrane: the alveolar wall, the alveolar basement membrane, capillary wall, the capillary basement membrane, and then we're going to have oxygen. Sorry, we're going to have oxygen over here. That's going to diffuse from the lungs into the blood, and we're going to have carbon dioxide over here. That's going to diffuse from the blood into the lungs, and that's the gas exchange. Okay, so we will get into more detail on this and the rest of respiration next week. Okay, we will redraw this, but I'm going to take it down for now so I can see you guys better. Any questions on that terminology? Or anything else that you want to talk about? So we have Monday to finish chapter 7, which will be a lot of information. Please don't miss that lecture. A lot of information, a lot of very important information in that lecture. And then your review and then your exam. So it's coming up. So please keep up. If you've fallen behind, please put in some time over this weekend to get caught up. Okay, you guys have any questions for me? Okay, I'll stay on for a couple minutes if you want to stay and ask a question. Otherwise, have a great weekend. I'll see you on Monday.
Thanks, Professor.
You're welcome.
Professor, I have a question.
Go ahead.
For chapter 6, can you explain again the 3 ways that decrease peripheral resistance?
Yes. So look at the formula for what's the formula for peripheral resistance?
It is length times viscosity divided by radius 4.
Okay. So if it's asking, is the question asking for a decrease in peripheral resistance or an increase?
Name 3 ways to decrease peripheral
Okay, so we're
resistance.
trying to decrease peripheral resistance. So the 3 things that affect peripheral resistance are length, viscosity, and radius. So what has to happen to length to get a decrease in peripheral resistance?
It has to decrease?
Decrease in length. What has to happen to viscosity?
An increase?
Length and viscosity are directly related to peripheral resistance, which means they move together. One goes up, the other goes up.
Oh, so they'll both decrease?
So a decrease in length would cause a decrease in resistance. A decrease in viscosity would cause a decrease in resistance. Radius has an inverse relationship. So what has to happen to radius to get a decrease in resistance?
An increase?
An increase in radius. So those are your 3 things.
Oh, okay. Thank you.
So for external control of blood flow, is it just epinephrine causes vasoconstriction everywhere except near the skin?
That's all we talked about. Yep. And you should mention the arterioles there. Epinephrine acts on the arterioles to cause vasoconstriction.
I have a question.
Roxanne, go ahead.
So I missed like the first lecture, and I've been trying to kind of catch up ever since. For the cardiac auto conduction being the intrinsic control of heart rate, right?
Mm-hmm.
What am I missing? Because I got like the SA node sets the beat to 100 beats per minute. And then, okay, I wrote it down, now forgot it. But what all am I missing? What components?
So I gave three functions of the SA node. One was to initiate the signal, because the signal's not coming from the brain. The signal's coming from the SA node. It initiates the signal, it sets the rhythm at 100 beats per minute and causes contraction in the atria.
Okay, so that's it. That's the only thing. And then do I need to, like, would I need to, if I were explaining that, include that the SA node sends the signal down to the AV node and then sends it to the bundle?
You gotta go through the whole thing. Yeah, the AV node, then the signal goes to the AV node, which delays the signal so we can finish filling the ventricles. Then the signal goes down the AV bundle to the Purkinje fibers, which contract the ventricles. Yeah, you explain the whole thing.
Cool, thank you.
Mm-hmm. Guillermo, you wanted the two formulas for blood pressure?
Yes.
Okay, so it's just a short version and expanded version. So short version is blood pressure equals cardiac output times peripheral resistance. But then we can expand cardiac output and we can expand peripheral resistance. So if we expand the whole thing, it ends up being blood pressure equals heart rate times stroke volume, which together makes up cardiac output. And then length times viscosity over radius to the fourth, which makes up peripheral resistance.
Oh, okay. Oh, thank you.
You're welcome.
I have a question. Oh.
David, go ahead.
Are we supposed to know how many O2 molecules are consumed during the oxidation of a carbohydrate?
No, I didn't teach that. I won't ask it.
Okay.
Professor, can you go over the intrinsic of blood flow and the extrinsic of blood flow?
Sure. So if we're talking about controlling blood flow, we're going to be talking about the arterioles. The arterioles control blood flow through vasodilation and vasoconstriction. So remember, extrinsic control is something else, something external acting on it. So the only thing external we talked about acting on the arterioles is epinephrine. So extrinsic control of blood flow is epinephrine acting on the arterioles, and that causes vasoconstriction everywhere except at the vessels near the skin. Vasodilation we talked about happening at the exercising muscle in response to the local conditions. So that's when we listed those 6 conditions at the exercising muscle, sensed by the chemoreceptors, which triggers vasodilation.
Thank you.
You're welcome.
Professor, what did you say was the definition to Boyle's law again?
Boyle's law, it says there's an inverse relationship between pressure and volume.
Okay, thank you.
Mm-hmm. Sorry about that, Marcia.
Oh, sorry.
Go ahead, London.
Can you go over the definitions of the tidal wave? Like all the definitions, I'm sorry.
All of them? Okay, that's fine.
Okay, well,
yeah, I can do it. It's not a problem. Not all of them have definitions. Some of them just have formulas.
Formulas, yeah, okay.
So tidal volume is the amount of air moved through your lungs in a normal breath. Inspiratory reserve volume. Inspiratory reserve volume is the amount of air you could inspire beyond your normal breath. Okay, the term kind of explains itself. It's your inspiratory reserve. It's what you could, it's what you could inspire extra. And then inspiratory capacity is the combination of those two.
Wait, Professor, I can barely hear you. Let me fix my. Hold on. Oh no, this is.
Can everybody else hear me? Okay.
I don't know why I can barely hear you on my end. Okay.
You can also send me an email or send one of the PLFs an email, and they can, or somebody else in the class, and they can give you that information.
Okay.
Okay. All right. Anything else, guys? All right. Have a great weekend.
Thank you, Professor.
You too.
Thank you, Professor.
Bye.
Thank you.